Good afternoon, everyone. My name is Barbara Rae-Venter, and welcome to Solved: Unmasking the Golden State Killer. Whoops. So my book is I Know Who You Are, and this was published in just about 2 years ago by Penguin Random House. I have 2 book signings that are scheduled, one this afternoon at 5 o'clock and then one tomorrow at 5:15. So in my book, I cover some of the early cases that I worked on. There are 6 cases there. Of course, the Golden State Killer, Lisa Jensen, and I'll be mentioning her briefly in a little while. She was a woman who was abducted as an infant, and so she was actually the first criminal case that I worked on. Then we have the Clearfield rapist, and then 3 unknown, or at least unidentified remains cases: Baby Doe Seattle, the Billboard Boy, and Precious Jane Doe. And then today I'm just going to be covering Golden State Killer. So since the theme for the seminar is families, I'm going to talk a little bit about some of the other family-type cases that I've worked on, because, of course, in trying to identify suspects in these criminal cases, what I'm doing is I'm using the family histories of the people who share DNA with whoever a suspect is. And so along the way, of course, we've ended up, some of the people who share DNA are people who are adoptees. And so we actually end up then having to solve who they are before we can solve our case. So I actually got started in doing this when I was working on my own family history, and I'll talk about that a little bit. And so there are some people that were important there. A guy called David Abbott, who lives in Kent in England. For those of you who've read my book, you will know who Patrick Roundtree is, and there's a little story that goes along with him. Then there's a couple of adoptees. Adam Keim, who actually was helpful in solving the Lisa Jensen case. So he was an adoptee. He was one of her closest matches. It means he shared the most DNA with any of the other people in the database. And so he's one of the people that, we had to solve who he was in order to help solve who Lisa was. Alan Law is an interesting case I do some adoption work with a group called DNA Adoption. And Ellen Law had learned when she was in her 80s that she'd been adopted. And so she very much wanted to learn who she was. And so she and her granddaughter had gotten a copy of her birth certificate. She was born in Chicago, but the names on the birth certificate had been falsified. And so they'd spent basically 10 years trying to find these people, not knowing that these were not true names on her birth certificate. And so when she came to DNA adoption, she was 98 years old. And so normally we would tell people to take our class. In this case, I decided, well, she might not make it to the class. So I agreed to help her. So it was a very sweet story. Then we'll talk a little bit about investigative genetic genealogy. And there again, Lisa Jensen was an abductee. She was abducted when she was about 6 months old. And of course, we'll talk about the Golden State Killer. So what is genetic genealogy? It's the use of DNA to basically to assign biological relationships to the interactions between 2 people. So if you're trying to do your family history research, how many have you done DNA testing? Oh, good, a lot of you, that's great. So you know that you get a long list of people and how much matching DNA they have with you, and then you need, of course, to try and sort out how they actually are related to you, because quite often you get people that you've never heard of that share DNA with you. Then investigative genealogy or genetic genealogy is just basically taking the same kind of information, census records, birth and death and marriage records, and so on, and then using that to identify somebody who is a suspect in a violent crime. And of course, the amount of DNA, matching DNA, is expressed in centimorgans. The more centimorgans you have in matching with somebody, then the more closely related they are to you. And so we use tools like this one. This is a chart that was put together by a guy called Blaine Bettinger. So this is citizen science at work. This chart he compiled from 52,000 people who sent in their DNA matches with known cousins from their own work that they'd done with either Ancestry, 23andMe, or Family Tree DNA. And then he compiled this chart to get an idea for how much DNA you share with various cousins. And so we use this information then to try to figure out how people are related to our suspects in our criminal cases. So the DNA timeline has actually moved fairly quickly. The seminal paper on the structure of DNA showing that it was a helix was published in 1953 by Watson and Crick. 50 years later, the Human Genome Project was completed. I should put that in quotes because actually it isn't quite complete, but for all intents and purposes, it was complete. And then fairly quickly on the heels of that, there were some direct-to-consumer tests that became available for testing mitochondrial DNA and Y-DNA. So the mitochondrial DNA, there's a little organism or organelle in the cytoplasm of your cells called the mitochondrion, and it has its own separate DNA. And it's kind of interesting because it's passed down the maternal line. So a woman passes her mitochondrial DNA onto all of her children, but only the female offspring actually pass it on to the next generation. For those of you who've done any biochemistry or remember your biochemistry and you remember ATP, the mitochondria are where the ATP is produced. And it's been kind of interesting. One of the symptoms of people who had long COVID was tiredness. And it's expected that probably what has happened is that the virus was actually damaging the mitochondria. And so people were not producing as much ATP as they needed, and so they felt really tired. Y-DNA, of course, only men have a Y chromosome, and that can be used for tracing the direct paternal line. Then around 2009, the type of DNA that we use for doing our forensic cases is autosomal DNA. That's chromosomes 1 through 22. I'll show you a little bit. So this is, I promise there are only 2 science slides. So this then is showing the human karyotype. So we have 22 sets of chromosomes, or the so-called autosomes, and then we each have a set of sex chromosomes. In men, of course, it's an XY, and in women we have 2 X chromosomes. So you inherit one member of each of those pairs from your father and one from your mother. And for the sex chromosomes, of course, men inherit an X from their mother and a Y from their father. Women inherit one X from their father and one from their mother. All right. This is to try and show the different kinds of DNA that we're working with when we're working on these cases. People tend to think there's only one kind of DNA. There's actually several kinds. So we already talked about mitochondrial DNA and autosomal DNA. Now within the in that we've also got then the Y chromosome. And so if we now look at this and we're going to try and trace where different kinds of DNA come from and how they can be used, let's go through this. So we've got, is this my pointer? Oh, whoops, that isn't what I wanted to do. I don't think the pointer's working. Oh, well. So let's start with Sunny here. I think the problem is my nails are too long.
Oh. I wish I could do it for you.
Yeah, that's not going to work very well. Okay, so let's take, so his Y chromosome is in blue. And so you can follow that up. I'm sorry, I'm not going to be able to work the pointer. My nails are a little long. So anyway, so you can follow it up. He's inherited that blue chromosome, the Y chromosome, from his father, going up the left here, who's inherited it from his father, and of course from his father. So you can use the Y chromosome then to trace your direct paternal line if you're doing family history research. It's inherited virtually unchanged from one generation going back many, many generations. You do of course get mutations along the way, and in fact my brothers have one mutation between them. So the older of my two brothers, his Y-DNA is identical with my dad's. The younger brother, he's got one mutation, which was kind of interesting. The mitochondrial DNA is the circle. It's a two-stranded circle of DNA, and that comes in down the maternal line. As I indicated, the mitochondrial DNA is passed on by a woman to all of her offspring, so Sonny Boy gets his mom's mitochondrial DNA, but he's not going to be able to pass it on to his children. His sisters, on the other hand, would. So you can, again, go up each of the generations, and you can use it to trace the direct maternal line. Again, it's inherited virtually unchanged from one generation to the other. Mutations do, of course, occur, but for the most part, you get the identical mitochondrial DNA that your mother had. The more complicated is the autosomal DNA, and it's because it's complicated that it's so powerful for tracing things like suspects in violent crimes. The autosomal DNA goes through a process called recombination, where the DNA that you inherited from each of your parents recombines at the point of making either sperm cells or egg cells. And so if you look at, on Sonny, we've got that bright yellow strip of DNA. You can follow up on here as to where it originally came from. So you can see he inherited it from his father's father. He inherited from his father's mother, and then she then inherited from her parents up there at the great-grandparent level. So this becomes important because if you are matching with somebody as a second cousin, what do you know about those people? You know that you're sharing a set of great-grandparents. So if we were to look at this and decide that Sonny has matched with somebody else in one of the databases with somebody as a second cousin, what do we know about them? We know that if they're sharing that yellow segment of DNA, that they're sharing that set of grandparents or great-grandparents up on the top left there. And we know a little bit more. We know which member of the pair, but just for sake of discussion, we know that they're both descended from that set of great-grandparents. If we now add a third person here who's an unknown, but they're also sharing with Sonny and his second cousin that yellow segment of DNA, they then all three of them then are sharing that single set of great-grandparents. So what do we know about our unknown person? They must be a descendant of that set of great-grandparents. So if we look at it in a slightly different format, again, we've got Sonny down here, we've got his second cousin. We're then showing that they've got a set of great-grandparents. If we then had the third person in here who's the unknown, what we would do is we would now turn around and build what's called a reverse tree, and we would look for all of the descendants of that shared couple in the diagram here, because we know that those three people all share that one set of great-grandparents, and they're all descendants of that set of great-grandparents. So I retired in 2005, at least I thought I had retired. And so one of the things that I wanted to work on was my own family history research. So this is then part of my family tree going back to my great-grandparents. And so my mother, so for those of you doing research in the family history, you want to try and identify who the immigrant is in your family. So in my case, on my mother's side of the family, my mother was actually the immigrant. She was born in Newcastle upon Tyne in England, and she and her family had emigrated to New Zealand in 1913. And so my grandfather, her father, was actually born in Scotland. I didn't actually know anything about my father's side of the family. For whatever reason, either he didn't know or he really just never talked about it. And I had always assumed that they were also just off the boat from Scotland. So I started researching people, and what I discovered was in 2006, my parents were in a car accident. My father was driving, he had a heart attack. My mother was killed in the accident, and then my dad actually spent many, many months in the hospital being put back together again. So I was staying in their home, and my dad had asked me to pay bills and so on, so I'm going through his desk, and I discover that his mother, so Eleanor MacLeod up there, she was actually born in New Zealand. I was astounded, I had no idea. And not only that, according to her birth certificate, her father was also born in New Zealand back in 1862. So this was totally new information for me. And he was born in a place called Waipu, and I'm going to show you a little bit about what I then found out about that family. Meantime, let's take a look at some of the individuals that I discovered on here. Some of them are people I would really like to know more about. So let's take a look then. I started building out then, of course, my father's line. I discovered they were all born in New Zealand too. So this was really a surprise. So I get back to Charles Joseph Rae is actually the immigrant, the guy all the way on the top right there. And he turned out to be a very interesting person. Unfortunately, I can't find him. I lose him in London, which is not a good place to lose people. By trade, he was a painter and a paper hanger. So somebody who, would help you refurbish your house. But he was a prolific writer, very involved in politics, set up the labor unions in New Zealand. And on top of that, it mentions in the top quote there that he helped in establishing the Mechanics Institute in Christchurch in New Zealand. The Mechanics Institute was the forerunner of the library system for Christchurch and then ultimately for the rest of New Zealand. How did a guy who was a painter and a paper hanger, where did he get the education to be doing all of this incredible stuff? He was apparently a very eloquent speaker and very powerful writer. So somebody that I would definitely like to learn more about. Oops. Another interesting person in going through my mother's line. So my mother was the family historian, but she really didn't know what had happened to her father's siblings. He had two brothers and a sister, and really couldn't find very much on them until the 1911 census was issued in the UK, and I was then able to find his siblings. One who was of particular interest is this one, Alan Grant. And Alan Grant was a very interesting guy. He was an inspector in the London Metropolitan Police. And of course, there's a very famous case of a serial killer in London around that time period, Jack the Ripper, who was active in 1888. My great-uncle would have been a little bit young when he actually was active, but I would guess that he probably worked on the case at some point, because he did join the London Metropolitan Police when he was in his early 20s. What was interesting also about this fellow is that when I started researching his records, I was able to get his actual personnel file from the archives in London, and it turned out before he joined the police force, he was a rabbit catcher. So he worked on some lord's estate catching rabbits. Now he was catching criminals. One of the families that actually got me into doing a lot of research, well, actually, let me back up. So there was one other, so for those of you who've actually read my book, I have a story in there about somebody who turned out not to be who they thought they were. That was My cousin, he's a Roundtree cousin, so this was, my grandmother was a Roundtree. So this was the son of her youngest brother that I actually got to do DNA testing for me. He was in his 90s and didn't hear very well, so I couldn't really talk to him on the phone, so I actually arranged all of this through my cousin, who was my age, for him to do DNA testing for me. So I was really excited. My grandmother was a Quaker, and the Roundtree family was a large philanthropic family in Yorkshire in England. And so I was very interested in seeing, what the connection was with the family that was in Yorkshire. My part of the family was actually in Sunderland in England. So I get John Patrick, who was my cousin's name, so John Patrick tests, and he came back matching with hundreds of people in Clan Irwin. He didn't match with any Roundtrees. So I found two other Roundtree male cousins, neither of whom I knew. I found one in Wales who agreed to test. So he tests, he matches with John Patrick, he matches with all the guys in Clan Irwin. I then tracked down another cousin in Perth, Australia. We shared, I think it was a 6 times or a 7 times great-grandfather. And so I wrote to him and I explained what I was trying to do, and he said, sure, I'll help. What's one more illegitimate person in my tree? So he came back matching with John Patrick, the cousin in Wales, and of course all the men in Clan Irwin. Now, Clan Irwin was in Dumfries in the south of Scotland, and Sunderland, of course, is in the north of England. And the time period I was now getting back to was a period called the Border Reivers. And what this was folks who lived in the north of Scotland, at least the north of England and the south of Scotland. They would maraud across the border, stealing wives and cattle and what have you. And they actually developed what they called border law. And the border law allowed for you, if you went with all due hue and cry, you had to make lots of noise, go across the border. You could bring back your cow or your wife, whichever one you wanted, and there would not be any prosecution. But you had to do it within 3 days, and you had to do it with hue and cry. I'm suspecting that that's probably what happened with my Roundtrees, that there was a wee lassie that ran into a wee laddie from Dumfries, and that's why we now related to the Clan Irwin folks. An interesting question that I have about Clan Irwin is, maybe I'm related to John Irving. So these are my cousin's top matches. We've got an Irving, and then the other, these are variant names then for Clan Irwin. So we've got Irvings, we've got Irvines, and we've got Irwins. And you can see this is pretty close. They're testing 644 markers, and there's only 6 that are different between the top Irving match and my cousin Patrick Roundtree. All right, so the family, though, that really got me into wanting to use DNA for doing family history research was, of course, Eleanor MacLeod's line. Because my great-great-grandfather there, Donald MacLeod, was, as I said, he was born in Waipu. And this is how my ancestors got to Waipu. So they left the north of Scotland in about 1817, they were Presbyterians, following their pastor, the Reverend Norman MacLeod. There were about 400 of them who all left Scotland and went to Nova Scotia. Initially, they settled in Pictou, and then ultimately they settled in St. Anne's on Cape Breton Island. They were there for about 3 decades. They did very well, and then unfortunately, potato blight and wheat blight contaminated their crops, probably coming in from some of the ships from Europe. And so they're basically starving. So the Reverend had 3 sons who decided to leave and go to Australia. And so they went to Adelaide in South Australia, and they wrote back and said, come on over, lots of land, it's just great here. So what they did is they built 6 ships, And then over a period of time, they started out on their trip from Nova Scotia to South Australia. So of course, the Reverend went on the first ship, and unfortunately, when they arrived in Adelaide, it was the beginning of the gold rush. There were whores and booze. These Presbyterians were very unhappy. So the Reverend wrote to the governor of New Zealand and said, do you have any land you can give us? We have about 1,200 people. And so the governor of New Zealand wrote back and said, yes, you can have some land in Waipu. So then they continued on to Waipu, which is in the very north of the North Island of New Zealand. They're at number 9 on the map there. And so my great-great-grandfather then was born in Waipu, as I said, in 1862. All right, let's talk about some of the cases. So one of the things that happened is when I started working on my cases and using DNA, is I started getting matches with a lot of people who were adopted. And I had absolutely no clue how to help them. So what I did is I went online and I found a class that I could take. David was interesting because I was his biggest match. He had learned, he was watching, I think it was Find Your Roots on television, and he's in his 70s, and he thought this sounded really kind of fun to do some family history, and so he actually did a DNA test with Family Tree DNA. And then he wanted to try to find out some information about himself. He needed his birth certificate to figure out when he was born and so on. Of course, he thought he knew when he was born. But he went online to get his birth certificate. I'm not sure how you get to 70 years old without needing a birth certificate, but anyway. And he discovered that he couldn't find himself in the database. So on a hunch, he put in his mother's maiden name, and bingo, there he was, and he was a year older. He had been born a year before his mother married the guy that he thought was his father. So here he is, learning in his 70s that the man he thought was his father was not, in fact, his father. And so that was sort of where I came into it. Unfortunately, I matched him on his maternal side. He had his mother's sister was still alive, and she tested, and so I matched with her. But he was really one of the first people that I tried to help, and it was because of him that I went and took a class. So the class I took was done through DNA adoption. It was 6 weeks long, and lots and lots of new information. And then one of the things that happened is that the folks who were running the class discovered I had a science background, and so they asked if I would help teach the classes and answer some of the webmail that came in. So I agreed to do that, and then this is Ellen Law, and so she's the 98-year-old. So I helped Ellen. She was one of the first people that I actually helped rather than tell them to take a class. So this was really kind of fun, and it illustrates how important family is to people. So because she was so old, she actually had a number of very, very close matches on 23andMe. And so I was actually able to very quickly figure out who she was. The phony names on her birth certificate were actually similar to what the real names were. And so when I'm putting it together, I could say, aha, yeah, this is the right person. So the people that I then identified as being her family, they lived in Minnesota, and they flew out to San Bernardino to meet her. And they sent me a ticket, which was really nice. What was amazing is the woman just to her right, who's listed, Kathy Castillo, who's listed as being her niece, her mother was actually Ellen's full sister. It turns out after Ellen had been given up for adoption, her parents actually married. And so she actually had 2 full siblings that she didn't know anything about. So as you can see, there's just an amazing resemblance between Ellen and then the folks on the right there who are the cousins from Minnesota. She was just so thrilled. I mean, you can see on her face. It was just so important to her to finally be meeting people that looked like her and that were her really close relatives. So this was a really fun reunion. So then there was a webmail that came in from a Deputy Peter Headley in San Bernardino, California. And what he wanted to know was the technique that we were teaching to adoptees on how to use their autosomal DNA to find their birth relatives. Could that be used to identify a woman who had been abducted as an infant, now in her 30s, had no idea where she was from or anything about her family history? Of course, law enforcement wanted to know because they wanted to, find out who had taken her. But for her, of course, it was very important to try and figure out who she really was. So I said, yeah, the technique should work. It's exactly the same thing. You're just trying to find parents of somebody who doesn't know who their parents are. So we went ahead and we worked on this case. I was able to identify who she was. Her mother was a woman called Denise Bodan. She was from Manchester, New Hampshire, and then there were 5 brothers that I had identified. One of them would, of course, be her father. But it turned out her grandfather was still alive. So, of course, we asked her grandfather, so who is Lisa's father? Bob Evans, he says. Well, Bob Evans was not on my list of the 5 brothers. So on a hunch, the deputy sent a picture of the guy who had abducted Lisa back to the grandfather. Yes, it's Bob Evans, he said. So now we suddenly knew how the abductor had gotten a hold of Lisa. He'd actually been her mother's live-in boyfriend. So there's a whole extra story that goes on with that. I'm not going to go into that right now. Just to say that, obviously, the deputy was extremely excited that we had actually been able to identify who Lisa was, and so he kept telling everybody and their pet duck that we'd solved this case. So this was Lisa then, and so we solved that case back in 2016. This was, just to take a quickie, this was Adam Keim, who was Lisa's closest match. He was the adoptee that I then identified who his relatives were. And this is Adam at my book signing in San Francisco at the Commonwealth Club a couple of years ago. All right, the Golden State Killer. So he was actually thought to be 3 different people. He started out as the Visalia ransacker. And what he would do is he would apparently put people's homes under surveillance, and then when they were gone, he would go in, and he particularly liked ladies' underwear. He would dump out their lingerie drawer on the floor. He would steal little trinkets. He particularly liked taking a single earring. How irritating is that? He'd help himself to some food from the refrigerator, maybe have a beer, and then he would leave. So he did that to over 100 homes in the Visalia area in Northern California. He then escalated to rape, and he then became known as the East Area Rapist. The initial rapes were all in the east of Sacramento. And from there, he escalated again to murder. And he then became known as the original Night Stalker. Michelle McNamara was working with one of the analysts who was working on this case, and she's the one who actually coined the term the Golden State Killer when it was realized after about 25 years in DNA that actually these 3 personas were actually all one man. So this is just a map showing the home invasions, over 100 of those, up around the Visalia area, showing the rapes, and these absolutely terrorized the area. This guy was a sadist. He, after raping somebody, he would call her again. And this could be weeks later, months later. In one case, it was years later. This woman had actually gotten divorced, remarried, moved, and he called her. And what he would do is he would sort of talk through clenched teeth and say, "I'm going to kill you." So these cases, by the time that I started working on them, these are 40-year-old cold cases. These people have been terrified for 40 years that this guy is going to come back and find them again. So what he started doing is he obviously extended out from Sacramento down towards Walnut Creek and Pleasanton, and then on the other side down towards San Jose. He then moved south to just north and just south of Los Angeles, where he then committed 12 rapes. One of the rapes was actually in Northern California. So as I said, Deputy Headley, that I'd been working with, he had been telling everybody we'd solved this case. So at some point I got a phone call from Paul Holes and Steve Kramer. Steve Kramer was with the FBI, and they were working on the Golden State Killer case. And so they called me and asked if I thought I could solve who the Golden State Killer was using the same technique that I'd used with Lisa. So I agreed to help them, and our first problem was finding some DNA. Even though there'd been 50 rapes, we're talking a 40-year cold case. And so each time that there had been advances in technology, the detectives working on these cases, of course, would take some of their DNA and try out the new technology on it. In Paul's case, there had been 3 rapes in Contra Costa County, which is where he was. He'd used all of his DNA. And so hand in hand, he and Steve Kramer went around to the various counties. There were 11 counties where there had been rapes. Some of them still had DNA, but they didn't want to part with it. Others were like Paul, they'd used up all their DNA. One of the last places they contacted was Ventura County. Ventura County said, "We'll check and see what we have in the freezer." What was exciting about Ventura County is they had a medical examiner who did things a little differently. Instead of just doing 1 rape kit, he actually took 2 rape kits. One of them he would use for doing whatever tests he needed to do. The other kit was still sitting in the bottom of the freezer. It had never been touched. It had been sitting there for 38 years. So, with fingers crossed, DNA was extracted. It was almost intact. It was, we do what's called a call rate to see how the DNA is. It came back with 98%, so that meant there'd just been a tiny amount of degradation for this DNA in all that time. So we finally had, one of our other problems was that there was no place that could do the SNP profiles like Ancestry and 23andMe and Family Tree DNA do. Back then, they were done on, if you did a buccal swab, then you could have your SNP profile prepared. But there was nobody that was doing it from forensic samples. So we actually had to talk to Bennett Greenspan at Family Tree DNA, who said, absolutely, we'll help, it's the right thing to do. And so they then prepared a SNP profile for us. So we uploaded that to the databases in Family Tree DNA and GEDmatch, which is a third-party site where people who've tested any place can upload their file and then look for other people who've also uploaded there for relatives. 14th of February, the algorithms are running to see if we've got any matches. And of course, for those of you who've done this, you know that it used to take a little while for the results to show up. And of course, we're all logging on a gazillion times today to see if we've got a gazillion times a day to see if we have any matches. So what were we waiting for? We wanted at least a second cousin match so that we could build out our trees and figure out who the Golden State Killer was. So what were the odds of actually doing that? Well, they're not too bad. There was about a 58% chance that we would get a second cousin match. Much higher chance that we would get a third cousin match. That's if the database was fairly robust. If it was even better, so if you were doing this in, say, Ancestry, which is a much, much bigger database, then of course the odds of getting a second cousin match would have been a great deal higher. So while I'm waiting, I have to do something. So I ran the GSK's DNA through a website called Prometheus, which will tell you something about some of the characteristics of the person. So for example, it told me that he had blue eyes and that he was likely to be prematurely bald. There was also used to be a utility on the GEDmatch site, which also would estimate eye color. Again, it came back telling me that he had blue eyes. Oops. This is just showing the size of the databases. Back when we were doing this, they were a lot smaller than they are today. So what I did, using the information that I had, I put together a little profile for the person we were going to be looking for amongst the descendants when we got to building our reverse tree. So based on when the first rape was in Sacramento, I figured he's probably at least born in 1956 or earlier, probably lived in Northern California. We knew he had blue eyes, likely bald. And then from the tools on GEDmatch, we could look and see what his bioancestry was likely to be. It was primarily North Atlantic and Baltic, and then some Southern Mediterranean. We got our matches back. They were horrible. The biggest matches were 62 and 60 on those two websites. So I had an account at MyHeritage, and so Steve Kramer says, well, let's upload to MyHeritage. Today, you can't do that. They've changed their terms of service. They will not accept forensic samples. Back then, there were no rules that said we couldn't do that, so we did. And there was our second cousin match. So, at the top, so on the left is the GSK. On the right at the top, we've got a match at 244.8 centimorgans. On top of that, she also had a daughter on there matching at, where is it? I can't read it. 138 centimorgans. So the 244 was just so exciting. What was even more exciting was when we then built out a tree, is there's the MyHeritage match on the top there. It goes up to a couple called Lyman Ritchie and Ada Seeley. And we'd actually built out a tree for the match that we got on Family Tree DNA. Down there, it was a 52 centimorgan match, which actually had managed to build a tree. And there was Ada Seeley and Lyman Ritchie. We had our most recent common ancestor. So now we could build a reverse tree to identify who the GSK was. So we did that, and I came up with a list of 9 men. And one of those 9 men, one of the theories on who GSK was that Paul Hulse had come up with is that because a lot of the initial rapes in the Sacramento area had been in new housing developments, that probably he was somebody to do with real estate. So maybe a broker, an investor, maybe somebody in construction. And so, as luck would have it, of the 9 men, there was 1 man who actually fit that profile. So the decision was made to get a consensual DNA sample from a close relative. And Paul actually discloses who it was. It was somebody who was a sister of this guy. Obviously, she didn't like her brother. So this woman tests. She's another second cousin match. So her brother is not GSK, but she's also got an X chromosome match. And if you remember, I said earlier Men only have one X chromosome. So the fact that she's matching with the GSK's X, we know that she's related on his mother's side. And so when we then look at our tree, we see we've got Ada Seeley. So then what I did is, and because of the X match, I was actually able to then eliminate 3 of the men who were on our suspect list. I was now down to 6 men. So what the FBI did is they took the driver's license records for those 6 men, and they looked at the eye color for the men. So over here in the column F there, we see we've only got one of those 6 men has blue eyes, and that was Joseph James D'Angelo. And then if we build out his tree, we can see, in fact, that, yes, he's a descendant of Ada Seeley. So, of course, the next step is to prove that he really is the Golden State Killer. So surreptitious DNA was collected. He was put under surveillance, and he went shopping at a mall. While he was in the mall, law enforcement swabbed the handle of his car door, tested that DNA, gazillion to 1 odds it could be anybody else. But since this case was so important and had been going on for so long, they went and got a tissue out of his trash and did it again, got the same result that, yes, gazillion to 1 odds it could be anybody else. And for any of you who actually watched the hearings after he was arrested, so the hearings for the case itself and then the victim impact statements, he would come in in a wheelchair looking totally catatonic. When they had him under surveillance, he was riding his motorcycle on the freeway at 100 miles an hour. They have videos of him doing burpees in his cell. It was all just part of the act that he put on. There we go. So he was sentenced in August of 2020 to 11 consecutive life terms. So he's not going anywhere. And so he's currently in the Corcoran State Prison in California, and hopefully he will be staying there. So this really showed the power of this tool. Paul Holes had been working on this case for 22 years. So the team of us working on this, so it was Paul Holes, Steve Kramer from the FBI, another FBI agent, Melissa Paraso, and then 2 people from the Sacramento DA's office. It took us 63 days to identify who he was. So let's look at some numbers. I'm hopefully going to remember all these numbers. So the case was 43 years old. There were 15 agencies that worked on this case. It involved 650 law enforcement officers and agents from the FBI. They collectively put in around 200,000 man-hours on this project. They swabbed 300 people for DNA. They had 8,000 people they put under surveillance at one time or another, and they spent $10 million. Number of suspects, 0. Investigative genetic genealogy. We spent $217 on a consensual DNA kit. There were 6 of us working on the case. We took 63 days, and we identified 1 suspect.